Hello and welcome to your online lecture for the course Altered Physiology 1. In today's lecture, we're going to be covering the second part of our immunity topic, looking specifically at hypersensitivities and immune deficiencies. We're going to start by looking at hypersensitivities, and I'll define hypersensitivity first. So hypersensitivity is an altered immunological response to an antigen that ends up resulting in disease or damage to the host. Hypersensitivity reactions can really be classified in one of two ways. One, by the source of the antigen that the immune system is attacking. So is the source allergy, autoimmunity, or Aloe immunity? But we can also classify it based on the mechanism that causes the disease. On this slide, we're going to be covering that first classification, which is that hypersensitivity reaction can be classified according to the source of the antigen that the immune system is attacking. Again, allergy, autoimmunity, and Aloe immunity. The term allergy refers to the deleterious effects of hypersensitivity to environmental antigens. It's really this exaggerated response to something in the environment. And so we call that exogenous, something that's occurring initially outside the body. Autoimmunity is a disturbance in the immunological tolerance of self-antigens, your own body cells. The immune system normally does not strongly recognize the individual's own antigens. Healthy individuals of all ages, but particularly older adults, may produce low quantities of antibodies against their own antigens, and these are called autoantibodies. And they can have this present without developing really an overt autoimmune disease. So we can say then that the presence of low quantities of these autoantibodies does not necessarily indicate a disease state. Autoimmune diseases will occur, though, when the immune system starts really reacting against its own self, against self-antigens, to such a degree that the person's own tissues are going to be damaged by these autoantibodies, or what we call autoreactive T cells. So there are many clinical disorders that are associated with autoimmunity, and they are collectively referred to as autoimmune diseases. Aloe immunity, also you might see this written sometimes as isoimmunity. This occurs when the immune system of one individual produces an immunological reaction against tissues of another individual. So you might wonder how that's possible. Well, Aloe immunity can be observed during reactions against, say, a transfusion that somebody has, or a transplanted tissue, or even the fetus during pregnancy. The mechanism that initiates the onset of hypersensitivity, whether it is consisting of allergy, autoimmunity, or Aloe immunity, is not completely understood, but it is accepted that genetic, infectious, and possibly even environmental factors are contributing to hypersensitivity. Lastly, when the immune system's response is not strong enough to protect the host, then the host is considered to be immune deficient. So these top three then are relating to hypersensitivity, kind of an overreaction, and immune deficiency would be an underreaction or not enough to protect the host. So let's talk more about hypersensitivity. And I should really begin by informing you that hypersensitivity reactions, they actually require what's called sensitization against a particular antigen that results in either a primary or secondary response. An individual will be considered sensitized when we could say an adequate amount of antibodies or T cells are available to cause a noticeable reaction when they're re-exposed to that antigen. Some individuals are going to become sensitized really quite rapidly, so after maybe having one single exposure to an antigen, whereas others require multiple exposures that may occur over years. Now, after sensitization has been achieved, hypersensitivity reactions can be considered to be immediate or delayed, depending on the time between re-exposure to the antigen and the onset of clinical symptoms. Reactions that occur within minutes to up to, say, a few hours after re-exposure are considered to be immediate, or termed immediate hypersensitivity, or an immediate hypersensitivity reaction. Delayed hypersensitivity reactions may take several hours to appear, and they are at maximum severity days after re-exposure to the antigen. Now going back up here under immediate reactions, the most rapid and severe immediate hypersensitivity reaction is a term you've, I'm sure, heard of, is anaphylaxis. Anaphylaxis occurs within minutes of re-exposure to the antigen and can be either systemic, so kind of generalized throughout the body, or cutaneous, so it's localized right in the area where exposure has happened. Symptoms of systemic anaphylaxis can include things such as itching, erythema, so redness, headaches, vomiting, abdominal cramps, diarrhea, problems with breathing, many different potential symptoms and signs. In severe cases, shock can result. So for example, systemic anaphylaxis includes a bee sting for somebody that's allergic to bees, peanuts, and fish as another example. Hypersensitivity reactions can be divided into 4 distinct types, which you can see listed on this slide. And we're going to spend the next few slides looking at each type and relating them to what we call a target antigen. So let me remind you of what I already mentioned, that hypersensitivity reactions can be classified in 2 ways. I mentioned this earlier, by the source of the antigen that the immune system is attacking, so allergy, autoimmunity, or Aloe immunity. So we spoke about this a bit already. And by the mechanism that causes the disease, of which there are 4 types: type 1, type 2, type 3, and type 4. Starting with type 1 reactions, they're also known as IgE-mediated hypersensitivity reactions. When you see Ig, it stands for immunoglobulin, and this is specifically immunoglobulin E, which is a type of antibody that is found in mammals. So this type of hypersensitivity is mediated by antigen-specific IgE antibody, IgE is the antibody, against environmental antigens. So it has to do with allergens. And it involves this release of the constituents of mast cells. And we've talked about the components of mast cells that can be released when the granules degranulate. You can see some of them in the image here. Allergies against pollen, food, and animal are typically considered type 1 hypersensitivity reactions. Let's look at pollen specifically. It's a very common allergy. When pollen is breathed in, we're going to look at the response that can occur. So here's pollen. Pollen has antigens on its surface that identify it as being what it is. And an antigen can be taken by a dendritic cell, which is an antigen presenting cell, where it will present the antigen to our immune system, to a T cell. So you can see it being sort of like almost handed off here. And then what's going to happen is the T cells are going to produce various substances that are going to lead to the production of IgE. If enough IgE is produced, it will bind to what are called Fc receptors that are found on different cells, but in this case, a mast cell. When it binds to the mast cell, if enough bind, then we're going to get an event occurring where the granules in the mast cell degranulate, releasing their substances, which can lead to a variety of different degrees of problems. There can be spasm of muscles, which in some people of the smooth muscle can make it difficult for them to breathe if their breathing tube, their trachea or their bronchial spasm. It can lead to swelling, infiltration of white blood cells, production of mucus, even damage to the epithelial tissue. And so because histamine plays a big role in this happening, when it happens to a large enough extent, it will signal that person to take an antihistamine to control some of the side effects that the histamine itself is creating. Now let's look at type 2 hypersensitivities, also called tissue-specific hypersensitivity reactions. These are generally reactions against a specific cell or group of cells, which would be a tissue. Cells express naturally a variety of antigens on their surfaces. And some of these antigens are called tissue-specific antigens because they are only expressed on the plasma membrane of certain cells. It's tissue-specific. It would probably make sense to you for me to say that the symptoms of many of these type 2 hypersensitivity reactions are going to be determined depending on which tissue or organ is going to express the specific antigen that ends up being the target. So let's have a look at how these reactions occur. The reactions are going to begin with an antibody binding to a tissue-specific antigen, or antigens that maybe have attached themselves somehow to a tissue. In any case, they're going to be the target of these antibodies. And what we'll see is that that cell that's being targeted can be destroyed by either lysis, which is sort of like shearing it, or it can just render it non-functional. There are 5 mechanisms of type 2 hypersensitivity, and it's shown in these images here. So we're going to go through them. The first one is a complement-mediated lysis. And what you can see happening here is a red blood cells being attacked by antibodies. And what happens with this is activation of the complement cascade through the classical pathway. And that's because we have involvement of antibodies. And so you might recall that from our in-class lecture. And so what ends up happening at the end of the complement cascade is we have this membrane attack complex that's formed, which punches holes in the red blood cell, causing water to infiltrate the red blood cell, causing it to be all sheared. And so that can happen in certain cases of blood transfusions, and we'll look at a similar example on an upcoming slide. In this next example, we can have antibodies causing cell destruction through phagocytosis by macrophages. And so you can see that happening here, where we have a red blood cell with the antibodies that have targeted it, and then the macrophage does what it's supposed to do and engulfs it, causing the cell to break up. In this next example, titled neutrophil-mediated damage, tissue damage can also be caused by toxic products that are produced by neutrophils themselves. So how exactly does this happen where neutrophils end up causing damage? Let's say there's some sort of antigen in our body. It could be something from an infectious agent or some of our own body's molecules that enter into our circulation. In some instances, those antigens can be deposited on the surface of some tissues in the body. And when those antigens bind to there, if there are antibodies for that antigen, then the antibody will bind to the antigen that's now on that tissue. And this is going to activate your complement cascade, resulting in the release of those complement factors C3 and C5. And we know that they are going to they're going to act as chemotactic factors for neutrophils. And so the neutrophils are just doing their job. They're going to reach the tissue and they're going to release any of their own mediators. Their granules are going to be released into the healthy tissue. And that ends up being toxic to the healthy tissue. Our fourth example here of tissue-specific hypersensitivity reactions is called antibody-dependent cell-mediated cytotoxicity. This involves natural killer cells. Natural killer cells in this example end up binding to an antibody on a target cell. This then causes the release of some substances that can destroy that cell, and the cell ends up dying by apoptosis. And in this last example, what we have here is the blockage of a normal receptor by an anti-receptor antibody, meaning that the receptors here should be open and available to, in this case, receive these molecules, which are acetylcholine. Antibodies can bind to and block those receptors, preventing acetylcholine from being able to attach to them. And this can occur with a condition called Myasthenia Gravis, in which the acetylcholine antibodies are blocking the acetylcholine from attaching to its receptors in that motor end plate of skeletal muscles, and thereby impairing the overall function of the muscle, leading to muscle weakness. So again, we have inappropriate production of antibodies causing these events. And when those events occur, destruction of tissue can occur because those antibodies, all they know what to do is to bind to antigens. This type of tissue-specific hypersensitivity is seen in the development of type 1 diabetes, but it's also seen with a condition called hemolytic disease of the newborn, and we're going to look at that in a bit of detail on this slide. So hemolytic disease of a newborn is also called erythroblastosis fetalis, and it has to do with Rh factor, which you might recall is present on some people's red blood cells. So if somebody has positive blood, then they have the Rh antigen. If somebody is negative blood, then they just do not possess that antigen. And you might also recall from first year anatomy that when we're born, if we're born with negative blood, we don't naturally have the Rh antibodies in our plasma. And that's relevant when we talk about hemolytic disease of the newborn. So let's look at this example where we have a male, and let's say he has, for simplicity, we're going to say he has A positive blood. And the mom here has A negative blood. And they conceive a child. That child could have positive blood, or that child could have negative blood. But let's look at an example where that child has A positive blood. So as I already mentioned, you're not born with antibodies against positive blood. So if you have negative blood, you don't have antibodies for positive blood that you're born with. So when this woman here is pregnant, there's going to be the movement of her blood through the placenta, and there'll be a very little bit that goes into the mom from the fetus. So overall, there is no issue. But once the mom delivers the baby, so the baby is delivered, there's going to be the exchange of blood, and this positive antigen on the baby's red blood cell is now going to enter the mom. So what happens when something foreign enters our body? Well, one of the reactions that our body will have will be to produce antibodies against it. So this baby's born, the positive blood enters the mom's blood, the mom reacts, the body reacts by producing antibodies against positive blood. And those antibodies will circulate now in the mom, and it doesn't cause any harm. They're just sort of like soldiers waiting around in case positive blood comes back into the body again. So from that first pregnancy, not a big deal because the mom didn't have positive antibodies floating around her blood. But now she gets pregnant for the second time. And it's going to be the same instance where dad's positive, mom's negative, and the baby ends up being positive blood. What will happen here now is the mom has these positive antibodies, these soldiers that are waiting to fight positive blood if it comes into the body. But in this case, it's a bit strange because those antibodies are going to be entering the fetal circulation just within mom's normal blood through the placenta. And those antibodies are now going to end up in the baby's blood. Now here's the problem. The baby is positive blood. The baby has A positive blood. The mom has just sent a bunch of antibodies into the baby's blood that are to fight positive blood. And so this is called hemolytic disease of the newborn, because what will happen, thinking back to our previous discussion on blood transfusions, this is sort of similar to that, in that it's going to cause lysis of those red blood cells. It's going to cause them to there's going to be an inflammatory state. It's going to cause them to infiltrate with water, as we talked about, and effectively burst. And that has a lot of potential problems for that baby. So one of the things that can be done to prevent this sort of thing from happening is knowing what the blood type is of the mother and the father, and also in particular knowing that if the mom is negative, we have to be very careful with her producing positive antibodies if she does have a fetus that has positive blood. And so there's injections that you can give to the mom to actually prevent this from happening, so that it's like every pregnancy is just like the first pregnancy. And so hemolytic is just the breakdown of red blood cells. So now we're on to talking about our third type of hypersensitivity, and this one is called immune complex mediated hypersensitivity reactions. Most of the type 3 hypersensitivity reactions are caused by an antigen antibody complex, sometimes called an immune complex. Okay, antigen antibody complex. And these are formed in the circulation and then deposited later in either vessel walls or other tissues. So you can see how that can be problematic. So in this image here, what we can see is an antigen and we can see an antibody. And they're going to join together and they're going to form a immune complex, and they can be of varying sizes. This one is bigger. This one is large. But what those immune complexes can do is then deposit themselves into tissue. And when they deposit themselves into tissue, it's going to create a reaction. And so the harmful effects of this immune complex forming and then depositing into the tissue is it's going to activate the complement cascade. And we're going to have those chemotactic factors that are released, which is going to attract neutrophils to the area. And so you can see these poor neutrophils are just, thinking they're doing their job, but they're destroying the body's own tissue. This is more commonly seen in the kidney compared to other organs, but it can really happen anywhere. These neutrophils will try to do their job and sort of attack the tissue because it thinks that's what needs to be attacked, but it's unsuccessful. But in the process, they release their lysosomal enzymes, their powerful digestive enzymes, which are going to then break down the body's healthy tissue. I should also mention that these small immune complexes usually aren't a big deal. They can be sort of cleared out by our circulation and don't cause tissue damage, but the intermediate size complexes and certainly the larger ones can adhere to healthy tissue, creating a type 3 hypersensitivity reaction. Now, just to finish up on this slide, in terms of the primary difference between what we spoke about earlier as a type 2 hypersensitivity reaction and a type 3 is that the type 2 hypersensitivity antibody is going to bind to an antigen that's on the cell surface. Whereas in type 3, the antibody binds to, we can say, a soluble antigen, so it's one that's just sort of present, but it's not bound to a tissue, and forming a complex which is then deposited into tissues. Now, to give you a clinical example of a type 3 hypersensitivity, you may have heard of celiac disease, which is a gluten hypersensitivity. And so following ingestion of the gluten, it can form those immune complexes that deposit into tissue, and the lining of the digestive tract can be destroyed. Now on to type 4 hypersensitivity. Type 1, 2, and 3, as you might recall, are mediated by antibodies. We talked a lot about antibodies. But T4 hypersensitivity reactions are instead mediated by T cells or T lymphocytes, so not involving antibodies. Type 4 mechanisms occur either through cytotoxic T cells or lymphocytes or your helper T cells. And how they do this is the cytotoxic T cells are going to attack and destroy the cellular targets directly. And that's the thing with the cytotoxic T cells, is they're very, very active in what they do. So they're the ones that are going to destroy the tissue. But the helper T cells may be a little bit more passive, where they produce cytokines, remember those chemical signals, that recruit and activate phagocytic cells, in particular your macrophages. So in this example here, it's showing the development of type 4 hypersensitivity specifically to poison ivy. And in this case, when we look at the primary contact, there's going to be some activation of T cells and some memory cells, but there's not going to be any rash, what we call dermatitis, inflammation of the skin. But the second contact, now that the person is sensitized to the allergen, it's going to activate a type 4 cell-mediated reaction that causes dermatitis or inflammation of the skin. Clinical examples of type 4 hypersensitivity reactions also include graft rejection. So that would be where somebody is, maybe they have damage to their skin, and so they're given a skin graft. So a donated skin. It also includes the skin test for tuberculosis, if you've ever had a test for that to see the reaction as to whether or not you have TB or you've been exposed to TB. It also includes other reactions. I talked about poison ivy, but being in contact with certain metals as well. A type 4 component may also be present in some autoimmune diseases, particularly rheumatoid arthritis, where the T cells attack the collagen that's found in joints. But let's look just a little bit more about what's happening, the pathophysiology of these type 4 hypersensitivities. So we talked about how T cells are going to be stimulated. We're going to have these cytotoxic T cells that are going to directly attack the tissue, and then the T helper cells that are going to do things such as producing cytokines and recruiting macrophages. What do the macrophages do? Well, they're going to attach to the target cell and they're going to release enzymes. So this could be, you've been exposed for, let's say, I don't know, the 4th time to poison ivy. Macrophages are going to attach to the skin in that area where you were exposed to poison ivy and release enzymes and also reactive oxygen species. And this is going to potentially cause apoptosis of the target cell. So in this very busy diagram, we can see the cytotoxic T cells, the helper T cells that have brought in some macrophages to the area. And then we're going to be targeting the cell in this reaction, causing the cells to undergo sort of a programmed apoptosis. So please remember that on the previous slide, I had mentioned that skin grafts, rejection of them is a type 4 hypersensitivity. This is also true of transplantation of other organs. So if somebody rejects an organ, the primary mechanism of the rejection of these transplanted organs is a type 4 cell-mediated reaction. Okay, so that was quite a lot of information on hypersensitivities. Now we're going to look at immunodeficiencies. And an immune deficiency is going to be really just defined as the failure of the immune system or failure of, say, an inflammatory response to respond normally, resulting in a person's increased susceptibility to developing infections. They have a reduction in their self-defense mechanisms. They are more prone to opportunistic infections. Opportunistic infections are caused by microorganisms that typically are not problematic in the average person. But if somebody is immunocompromised, those pathogens those microorganisms take the opportunity to infect that host. So caused by non-pathogenic microorganisms that wouldn't hurt the average person that now suddenly become pathogenic in somebody who doesn't have a well-functioning immune system. These infections can recur, and clinically they can present very atypically. So let's have a look. We're going to start by looking at primary and secondary immune deficiencies and some of the differences and then the different types that exist. Most primary immune deficiencies are the result of a single gene defect. That's a term we learned about in our genetics lecture. Generally, the mutations are going to be sporadic rather than being something that they've inherited. The mutations occur before birth. However, the onset of symptoms can be later. So that's an important consideration. So they are considered to be rare and congenital. Congenital just means that it's something you're born with whenever you see that term. In terms of the different conditions, we have an example here, severe combined immunodeficiencies. And people that have this condition, they lack an immune system. And so you can think of the boy in the bubble if you've ever heard that term before. They just have no immune system. There's also deficiencies that people can have in B cells, deficiencies in T cells, in your complement system, and some of the factors that are involved for that cascade to appropriately occur. Because remember, inflammation is part of an immune response, and you need all the parts for it to function properly. And you can also have a deficiency in phagocytes. Secondary immune deficiencies are much more common, and they are considered to be acquired, meaning you're not born with it, but it's going to develop at some point after birth. Secondary immune deficiencies are complications really of other physiologic or pathophysiologic conditions. So for example, infancy, pregnancy, and aging can lead to immune system deficiencies, psychogenic and physical stress, malnutrition, the presentation of certain infections, for example, HIV and AIDS, which we'll talk about later, cancer, but also the treatment of cancer itself can lead to an immune deficiency. Burns, being on immunosuppressants such as corticosteroids. So corticosteroids are used quite frequently for many different conditions, and they will reduce inflammation in the body, but it also reduces your immune system. Somebody who has their spleen removed, splenectomy, can also cause immune deficiencies, and people with diabetes mellitus. So this is not an exhaustive list by any means, but some common examples. And of course, the degree of secondary immune deficiency is going to be quite variable among these and among some of the details, depending on the extent of psychogenic and physical stress or malnutrition, just for example. We can say then that these here are factors that can cause secondary immune deficiencies, whereas these are the different types of primary immune deficiencies. Now let's have a look at the clinical presentation, what some of the clinical manifestations will be when somebody has an immune deficiency. whether it's congenital, whether they're born with it, or acquired, what we see as sort of the clinical hallmark of immune deficiency is this tendency to develop unusual or recurrent infections, severe infections, and simultaneous infections. The type of infections can sometimes indicate the type of deficiency that's present. So, for example, with T cell deficiencies, people with T cell deficiencies are more prone to infections from viruses, funguses, yeast, and some atypical microorganisms. Whereas people with B cell and phagocyte deficiencies, they are going to have difficulty fighting microorganisms that typically require opsonization, remembering that term from our in-class lecture. People that have complement deficiencies, these are associated with increased infection from the following bacteria, including Neisseria meningitidis and Neisseria gonorrhea. So the effects of what you see and what's happening in the person's body can give you an idea of what type of deficiency might be present. We're going to talk about the pathophysiology of AIDS, which stands for Acquired Immunodeficiency Syndrome. This condition is a secondary immune deficiency, and it develops in response to a viral infection, specifically caused by human immunodeficiency virus, or HIV. The human immunodeficiency virus, what it will do when it's in the body is it infects and destroys the CD4 helper T cells. And these cells are necessary for the development of other immune cells that are important, such as plasma cells and cytotoxic T cells. So if we can't produce these cells, then we have quite the suppression of the immune system. So HIV then suppresses the immune system, and if it's not controlled and if it continues to get worse, then it can secondarily create this generalized immune deficiency by suppressing the development of other immune responses against other pathogens and other microorganisms that enter the body. So if it's not controlled, then it leads to this generalized immune deficiency, which is called AIDS. So if somebody does have HIV, nowadays, depending on what's available to that patient, you can prevent it from that person developing AIDS, which is a lot more of a serious situation. So with people that have AIDS, they are much more susceptible to, remember the term opportunistic infections, where microorganisms that are normally not problematic take the opportunity to cause severe infection in immunocompromised hosts. We're still on the topic of HIV and AIDS, and we will be speaking of that on that topic for the rest of this lecture. But this slide talks about some of the lymphocytes in our body, so the B cells and the T cells, and it summarizes their roles. But I wanted to point out a few things. One is what I mentioned on the previous slide, that your CD4 positive helper T cells, they're going to differentiate into your cytotoxic T cells and your plasma cells, which actually come from B cells. And so we kind of lose this pathway here when HIV infects the cells. So let's just have a reminder of some of the roles of our lymphocytes in our body. So the B cells are going to be involved in secreting antibodies into our circulatory system to fight infections. So the antibody will bind to a specific antigen on a target cell. Hopefully it is a bad cell that it's targeting, and the antibody should neutralize the target. Cytotoxic T cells, though, work a bit differently. They are very active and direct. They're not passive at all. They're going to directly be killing cells that are infected. And cytotoxic T cells, they have the CD8 protein on their cell surface, which you can see here. Whereas our T cells that are helper T cells have that CD4 surface protein, which is targeted by HIV. But what the helper T cells do is they're going to, we know, differentiate into other cells, but they are going to also coordinate the activity of cytotoxic T cells and B cells. And so this is what we lose with HIV infection. And subsequently, our B cells, we spoke about the plasma cells on the previous slide, and your cytotoxic T cells, because of the attack on the CD4 helper T cells. So just a brief summary of what happens with HIV infecting cells. Remember, its main target is going to be the CD4 positive helper T cells, and this infection is going to impair other immune activations that would come from that. So other CD4 helper T cells, the CD8 positive cytotoxic T cells, even impacting the production and function of macrophages, and the B cells. We spoke more specifically about the plasma cells. So you can see how a lot of our immune system is impacted with HIV, and if this gets out of control, then it could lead to a more generalized immune deficiency that we would see with AIDS. This is a slide you can go through on your own, just showing you some trends with respect to AIDS throughout the world. You don't need to know any of the details that are listed here, but it's designed to show you that it is a problem. So we're going to look at some of the clinical manifestations or some of the effects that can come from having AIDS. Looking here, starting at the top, severe weight loss and anorexia is a common side effect of having AIDS. Kaposi sarcoma lesions, those are the lesions that you can see in this image here. And it is considered a cancer. So with AIDS, you're more likely to develop cancers. Perianal lesions from herpes simplex infection, also a side effect of AIDS. Deterioration of a person's vision from what's called cytomegalovirus, which ends up causing inflammation of the retina, and that can lead to blindness. We talked about opportunistic infections, so we can have some of these pathogens: pneumocystosis, atypical mycobacteria, cytomegalovirus, toxoplasmosis, and cryptococcosis, which can all cause what we say are opportunistic infections within the body that a person that has a normal functioning immune system should be able to resist a severe infection. And so we talked already about Kaposi sarcoma. Other cancers that you're more likely to develop when you have AIDS include lymphoma of the brain. Lymphoma is, in this case, it's a non-Hodgkin's lymphoma, but this is where you have malignant or cancer cells from lymph tissue that form in the central nervous system, in this case listed as the brain, and then cancer of the cervix as well can occur. Let's have a look now at the structure of the Hiv virion. HIV is a member of a family of viruses called retroviruses. And retroviruses, if you're not aware, are these RNA viruses that can insert their genetic information into a host cell, and that's how it replicates itself. And HIV carries its genetic information in the form of RNA rather than DNA. A couple of important enzymes that will come up on later slides include reverse transcriptase. Anytime you see ACE, it's an enzyme. Integrase and protease. These play a big role in helping the virus achieve what it's trying to do. There are 3 layers or coats when we look at the structure of the HIV virion. It has an outer layer that is primarily lipids. It has a middle layer that's made of what's called a P17 matrix, which we can see here. And that's just a structural protein making up that layer. And it has an inner layer called P24 capsid, which you can see represented here in green. There are two main what are called viral, because they're a part of the virus, the HIV virion, two viral glycoproteins. These are like antigens on the cell. They're cell identity markers that say what the cell is. They're found on the cell surface. Glyco means it's partly a carbohydrate and then partly a protein. The two examples that we have here include our GP120 right here, and this one's a very important one because it's this glycoprotein on HIV that's going to bind to those CD4 positive helper cells, preventing them from being able to do what they need to do. And then we have, and we'll see what that looks like shortly, and then we have GP41, which is just a transmembrane glycoprotein that is also important in the ability of the virus to infect human cells. So what we can see here is that HIV, the structure is designed in such a way that it has specific viral enzymes, specific layers, as well as specific cell identity markers that allow or contribute to its ability to infect human cells. So they're vital for the pathogenesis of HIV. We're going to look at the initial infection process of HIV. And this here is the HIV virion. And this here is a plasma membrane of one of the normal cells in the body, specifically the CD4 positive helper T cell. And so you can see that the virus is going to attach to a receptor on the CD4 helper T cell. And you can see that it's bound here, and the specific spot that it's bound to on the HIV is the GP120 that we spoke about earlier. GP just stands for glycoprotein. And it's also going to bind to a chemokine receptor. This is going to allow it to fuse, the HIV virion, to fuse with the plasma membrane of the normal cell or the healthy cell and put its genetic code, the HIV RNA, into the cell. So that's how it infects it, by unloading its genetic code into the cell. Let's look now at the role that the enzymes play in HIV infection. Remember the few that were mentioned on a previous slide. Retroviruses, such as HIV, use a viral enzyme called reverse transcriptase in order to convert RNA into double-stranded DNA. So in other words, the HIV virion is going to contain RNA, but because of an enzyme it possesses, it can convert its RNA, its viral RNA, into a double strand of DNA and end up integrating it into our own genetic code. So using a second viral enzyme called HIV integrase, the newly formed DNA is going to be inserted into the human cell's genetic material, where it can remain dormant. And if it's dormant, it will remain present throughout the person's entire life. But if the viral DNA is activated, that's where things are a little bit different. What we end up with is translation of viral information. This can result in the formation of new virions. It can result in lysis and death of the infected cell, shedding of infectious HIV particles. But in order for the viral DNA to, or I should say, in order for the HIV virion's life cycle to be continued, it does depend on proteases, which are needed to process proteins that are used to promote the cell's life. Specifically for the capsid, which if we look closely here, you can see houses the genome of the HIV virion. So I'm going to just go over the life cycle of HIV, and then we're going to talk about how the life cycle is susceptible to blockage at several sites. So the first thing we're going to look at and remind you of is this is our HIV virion, which you can see at GP120 is going to bind with CD4. It's all on a helper T cell. It's also going to bind to this chemokine receptor. Then it's going to be integrated into the plasma membrane of the cell, where it's going to put its genetic code, the HIV RNA genome, into the cell. Then it's going to make its way to the nucleus of the cell. It's going to convert from RNA to DNA. It's going to enter into the nucleus where our healthy DNA is, and it's going to integrate itself into our genetic code. Then what's going to happen, remember processes such as transcription and translation, is we're going to take a copy of this. Here's our copied strand, the transcript. Remember mRNA from learning about transcription and translation. That transcript, that copy of this genetic code relating to the HIV, is going to leave the nucleus. It's going to start forming a different structure. Then it's going to leave the cell and travel to infect another cell. So what we're going to look at is that the life cycle of HIV is susceptible to blockage at several different sites throughout this process. One of the ways is through entrance inhibitors, preventing it. These are some enzymes that can prevent the entrance of the genetic information. There's also reverse transcriptase inhibitors. These are going to prevent RNA, HIV RNA, from becoming DNA. There's also integrase inhibitors. These would prevent the DNA, the viral DNA, from entering into the nucleus. There are also protease inhibitors, which would prevent the development of this new structure that's being formed. There is a regimen for the treatment of HIV, which is a combination of drugs that are termed antiretroviral therapy, and it's designed to block at all these different levels in order to hopefully keep it under control and preventing a person from developing full-blown AIDS. The clinical progression of HIV begins within weeks after infection. The person can experience symptoms of what's called acute HIV syndrome. And during this period, the virus is just progressively infecting T cells and other cells and spreading throughout different areas of the body, decreasing the number of those circulating CD4 positive helper T cells, and setting up shop in some of the lymphoid organs in the body. What we can see happening quite quickly is a sharp drop in the CD4 positive helper T cells. We then have this period of what's referred to as clinical latency. And during this period, the virus is replicating, and T cell destruction continues, but it's called clinical latency because the person is generally asymptomatic during this period of time. The person may develop HIV-related disease, which is under this term here, constitutional symptoms. A variety of symptoms of acute viral infection might be present during this time. Now, when the number of CD4 positive helper T cells is critically suppressed, as you can see happening here, the individual is becoming very susceptible to a variety of different opportunistic infections and cancers, with a potential diagnosis then of AIDS. So if we look at this chart here, with this large drop-off that happened in the number of cases of actually people developing AIDS from HIV and then death as well, dropped significantly. And the reason for that was because of the introduction of the antiretroviral drugs that I had mentioned on a previous slide. We're going to look at plasma levels of HIV, so the actual virus itself, versus antibodies, and what levels are present at what time. So what we see happening here initially, this line here is representing the viral particles in the plasma. So initially, at the very start after infection, there's this sharp increase in the viral particles, sharp increase, and then it drops down. And what is slower and takes longer to develop are the presence of antibodies. So we can see antibodies increasing later on. What we can also see is that with this big increase in viral particles, it drops down and remains down here, dormant, until it becomes active again. And so some important features is that from week, say, 0 to about 9 or maybe 8, we have this sharp increase in the viral particles that are present, but we don't have antibodies. And so serologically, somebody would be negative because we're looking for the antibodies. And the antibodies are not being produced until later. So they have the condition, but it's showing up as negative. The other situation can be that somebody is going to be tested, say, at the point when their antibody levels are increasing, where they would be serologically positive, but asymptomatic. And then there is the point where they are going to be symptomatic, say full-blown AIDS, where they both are positive for the disease, but also symptomatic. And so we can call this region here the window period, so the period between infection and the detection of your first antibodies. Now, when somebody would show positive is going to vary depending on how they contracted the virus. So typically that window is going to be upwards of 7 weeks if it's contracted through a blood transfusion, which doesn't happen as often anymore, and anywhere from 6 to 14 months after somebody's had sexual transmission. And so the reason for that difference in window period is because the amount of blood entering the body after sexual transmission is quite small compared to a blood transfusion, which would signal the production of antibodies a lot faster than it would if it's been contracted after sexual activity. Okay, that is the end of today's lecture on hypersensitivities and immunodeficiencies. Thank you so much for listening. Bye for now.